A virus is defined by subtraction. The chapter's whole job is to fix the object of study as the thing that carries a genome, but nothing else life requires. No metabolism, no energy of its own, no cell division. It borrows every function from a host and, uniquely, replicates by total disassembly, then reassembly from parts rather than by growing and splitting in two. That subtractive definition is the generative primitive. Life is self-sufficient. Replication. A virus is replication outsourced. Lawton's, FedEx, and UPS anchor sharpens the pair. The virus is the abstract entity. The virion is its physical package in transit. Everything downstream in the book, structure, replication, immunity, disease, elaborates what a genome must do when it owns nothing but instructions. A virus fails the definition of life on exactly one axis: reproduction. The rest of the checklist it nearly satisfies: a genome, evolution, biochemistry built from the same nucleic acids and proteins. But living things divide. A cell grows and splits, substance continuous across the boundary, whereas a virus is assembled from scratch, its genome merely handing a hijacked cell the instructions to manufacture fresh copies. Continuity of matter versus continuity of instruction, that single swap is what expels the most abundant biological entity on Earth, roughly 10 to the 31st virions, from the category of the living. Lawton's FedEx anchor carries it. The virus is the company, the virion, the truck. You never see the entity, only its shipped copies. A virus is a genome with a delivery problem, and every feature the chapter names is subordinate to it. The capsid is a few protein types repeated without end, a brick wall, precisely because a genome too small to encode much cannot encode a bespoke shell. Self-assembly follows for free. Frankel. Conrad's tobacco mosaic, virions reforming in a test tube. And the deep classification axis isn't morphology, but distance to messenger RNA. Baltimore's 7 classes are 7 paths. A genome must travel to become translatable. Message, double-stranded DNA nearest, negative strand RNA, and the reverse transcribers furthest. Shape protects. The path to message is what a virus is. Predict the rest from that one constraint. A virus is the minimal solution to one problem. Carry a fragile nucleic acid message across a hostile extracellular gap and rebuild copies from scratch inside a cell. Every structural fact follows by economy. The genome is tiny, so it encodes only a few proteins. So the protective shell, the capsid, must be one protein type tiled over and over, held by weak electrostatic and hydrophobic forces. So those subunits self-assemble with no added instruction, as with TMV reconstituted in a test tube. Message plus shell is the nucleocapsid, and optional membrane stolen from the host is the envelope. Classification then. Keys on exactly the 2 degrees. The design leaves free genome chemistry and capsid symmetry, either helical or icosahedral. A virus carries almost nothing, a genome and a few proteins. So this chapter about the cell is really the chapter about everything the virus lacks and must borrow. Obligate. Intracellular parasites is the whole thesis. Replication is not something a virus does. It is something it induces the host to do. Lawton tours the eukaryotic cell, the membrane, the ribosomes, the rough endoplasmic reticulum, the Golgi, the mitochondria, the cytoskeleton. But each organelle enters already labeled as a future hijack target. The ATP it will steal, the ribosomes it will redirect, the exocytosis it will ride out. The primitive to carry forward, a virus is pure information addressed to machinery it does not own. To predict any infection, first catalog what the cell already provides. A parasite that carries no machinery of its own is fully specified by its host's machinery, so a tour of the cell is itself a tour of the attack surface. This is why the chapter reads the eukaryotic cell as an inventory of borrowable functions. Ribosomes make the proteins. The rough ER to Golgi line folds and ships them. Mitochondria power the theft. The cytoskeleton hauls the cargo. The virus genome encodes only the deltas it cannot steal. Everything else cataloged here is toolkit already present. Consider its place in the book's arc. Before you can watch a virus replicate, you must know exactly what it will requisition. The cell described here is the stage every later infection walks onto. A cell divides by doubling itself and splitting in two. A virus does something categorically. It dissolves. The chapter enumerates 7 stages: attachment, penetration, uncoating, replication, assembly, maturation, release. They hide one structural move. The incoming particle stops existing as a particle. Its genome becomes pure instruction, the capsid discarded. This is the eclipse, the interval where 0 infectious virions exist anywhere. The host's own machinery then mass fabricates parts that self-assemble into hundreds of new particles. Replication is thus not reproduction by growth, but disassembly into information, then parallel manufacture. Predict the rest from this. Every stage exists only to move a genome across a barrier, run it as a program, and re-encapsulate the copies. A virus carries instructions, but no machinery to run them. Replication is therefore not something it does, but something it makes the cell do. Chapter 4's thesis is that this coercion is an obligate ordered pipeline. Attach, penetrate, uncoat, replicate, assemble, mature, release. Every stage is a normal cellular function turned against its owner. Attachment exploits receptors the cell needs for its own signaling. Entry rides endocytosis. Translation borrows host ribosomes. The generative move to predict how any virus works is to ask which host process each stage hijacks. Tropism then falls out for free. A virus infects exactly the cells displaying the receptor its first stage requires. The program is universal. The target is set entirely by that first handshake. Defense splits along one axis, speed against specificity, and you cannot buy both at once. The innate arm fires within hours. Its pattern recognition receptors read shapes no host. Molecule wears, double-stranded RNA, nucleic acid sitting in the wrong compartment. It is a generalist that never learns. The adaptive arm takes days. One clone tuned to one antigen, costly, exact, and it remembers. The chapter's move is that bees are not redundant, but sequenced. The fast generalist holds infection down and licenses the slow specialist. Strip the innate arm, and nothing survives long enough to be taught. This is layered defense under time pressure, a pattern found everywhere. Cheap and immediate buys the runway for costly and precise. It is also the selective landscape. Every virus evolves to slip. Defense against a virus is one control problem solved at 2 timescales. The chapter's argument is that the 2 arms are coupled, not merely stacked. The innate arm fires immediately because it matches conserved patterns, molecular shapes no host cells should present, viral double-stranded RNA, nucleic acid in the wrong compartment. So it needs no prior acquaintance with this particular virus. The price is that it cannot be specific. The adaptive arm is exactly specific and it remembers, but it must be built, so it lags by days. The load-bearing move is this: innate recognition does not merely hold the line. It instructs and aims the adaptive response. Specificity trades against latency. Wiring the fast layer to switch on the slow one is how the host buys both. Diagnosis is inference, never sight. A virus is too small to see, so its presence is read off a signal it leaves behind. 2 signal classes. Exhaust the space. Direct signals are the pathogen's own molecules, antigen, nucleic acid, or live virus grown up in culture. Indirect signals are the host's trace of it, antibodies, or the cytopathic wreckage in infected cells. The load-bearing constraint is time. Viral load peaks early, then vanishes as the infection clears, while antibodies take weeks to rise, so the 2 channels light up in sequence, not together. Which test can possibly work is therefore fixed by when you sample. Detection is a time-indexed choice between 2 complementary evidence channels. Every technique the chapter catalogs is one cell of that grid. The virus is never seen, too small for light. So every diagnosis is an inverse problem. Infer the agent from the traces it leaves, never from the agent itself. 2 trace. Families divide the chapter. The first is the virus's own molecular signature, its nucleic acid, its antigen, and the damage it inflicts on cultured cells. The second is the host's countersignature, the antibodies raised against it, which is serology. Which trace is legible is time-indexed. Viral load crests early, then vanishes as infection clears, while antibodies lag by weeks, IgM first, IgG behind. The diagnostic question is thus never whether it is there, but what is observable now, a moving system read through proxies whose window the clock of infection sets. The chapter runs on one inversion. The properties that make a virus lethal are exactly the properties you weaponize against it. This is Laughton's double-edged sword. A virus enters cells effortlessly, commandeers their machinery, and replicates, so you point that same competence back at itself at 3 stations on the infection timeline. Before contact, an attenuated or subunit vaccine hands the immune system, a harmless rehearsal, so memory is armed before the real pathogen arrives. During infection, an antiviral interrupts one step of the replication cycle. After, turning the tables, the virus becomes the tool, a gene therapy vector, a gene therapy vector. 3 moves, 1 primitive, co-opt the replicator's own efficiency. Predict the 4th instance, and you already have the frame. A virus that causes cancer holds no cancer weapon. It disables the cell's own division control circuit, the same brake and accelerator logic every carcinogen deranges. 2 gene classes govern the cell cycle. Proto-oncogenes press the accelerator, tumor suppressors, PRB and P53, hold the brake. Cancer is unregulated proliferation, transformation, and it arrives whenever the accelerator jams on or the brake fails. Oncogenic viruses take the second route. Their proteins physically bind and neutralize PRB and P53, so the checkpoint that should halt a damaged cell releases it instead. The virus is thus not a special cause, but one path into a shared failure mode, predicting that any agent capable of pinning those two proteins is carcinogenic, viral, or not. A virus that causes cancer is doing nothing viral about it. It is a carcinogen structurally identical to asbestos or ultraviolet light, differing only in that it carries its mutations in as protein rather than inducing them in DNA. Cancer is one failure mode, uncontrolled division, reached by many roads. The oncogenic virus is simply a road that arrives preloaded with tools to disable the cell's own division breaks. The tumor suppressors, PRB and P53, the same targets a chemical carcinogen would have to knock out by chance. Here is the load-bearing constraint. Infection is never sufficient. The virus supplies one hit in the multi-step accumulation toward the hallmarks named by Hanahan and Weinberg, a contributor. Never the whole cause. A pathogen whose danger is not what it destroys, but which node it destroys. HIV attacks the immune system's coordinator, not its foot soldiers. It infects the CD4 helper T cell, the node that licenses every adaptive response and depletes it slowly. The name carries this, lenti, Latin for slow. Collapse the conductor and the whole orchestra falls silent, so patients die of ordinary opportunistic infections an intact system would have shrugged off. As a retrovirus, it reverse transcribes its RNA and integrates permanently into host DNA. The invader becomes indistinguishable from self, which is why cure resists and only lifelong suppression works. The immunodeficiency is not direct viral killing. It is decapitation of the network's control layer. HIV is the pathogen that infects its antagonist's own command node, the CD4 helper T cell, the coordinator of immune response. So its depletion doesn't subtract one defender, it collapses coordination itself. That is AIDS, death by opportunistic infection the disarmed system can no longer refuse. Two structural choices make it intractable. As a lentivirus, it reverse transcribes its positive sense single-stranded RNA into DNA and integrates permanently into the host genome. No clearance, no cured cell, only a lifelong reservoir. And it mutates faster than any vaccine can bind. 30 years on, attack the controller, then become part of the host. The chapter's arc, from death sentence to managed chronic disease, rests entirely on that asymmetry. Hepatitis virus is a name cut by effect, not ancestry. 5 agents, A through E, pulled from 5 unrelated viral families, disagreeing on genome type, transmission route, and whether they persist, are grouped solely because each converges on one target, the hepatocyte, producing a single liver syndrome. This is a polyphyletic category defined by what its members do, not what they are, like raptor or weed. The payoff sits downstream. Because the shared trait is phenotype and the internals are arbitrary, the clinical picture alone cannot separate them. Identity demands a molecular probe of lineage, not a reading of symptoms. 5 viruses sharing no ancestry: HIV, HBV, HCV, HDV, HDB, HEV, scattered across unrelated families, collapse into one category by a single fact: they all inflame the liver. The grouping is convergent, not phylogenetic. The name index is in effect, not phylogenetic. What binds them is the shared clinical signature, jaundice, the bilirubin backing up when hepatocytes fail. It is the downstream consequence read backward into a class. Expect then no molecular coherence. Transmission routes, genome types, chronicity, all diverge freely because nothing at the sequence level forces agreement. This is the same move as carcinogen or diuretic, a class carved by what its members do to a target, never by what they are. A herpes virus never loses the immune war because it stops fighting it. It hides. The generative move is one large double-stranded DNA genome, running two mutually exclusive programs. The lytic program replicates and kills the cell to spread. The latent program parks the genome nearly silent inside a long-lived reservoir cell, sensory neurons for HSV, B lymphocytes for EBV, invisible to the immune system, expressing only what lymphocytes for EBV, invisible to the immune system, expressing only what maintenance demands. Reactivation flips the toggle back when conditions favor transmission. That is why one virus gives cold sores now and shingles decades later. Every human herpes virus is this single switch instantiated with a different reservoir cell. Latency, not virulence, is the design. Persistence, bought by disappearing rather than winning. A virus family defined not by what it does, but by where it hides permanently. The hallmark of every herpes virus is latency. Never cleared, the genome retreats into a host cell and waits, reactivating under stress. The chapter's compression is that the three subfamilies are carved by latency site, neurons, immune cells, B lymphocytes. So the taxonomy is a map of hiding places, and a shared hideout predicts shared genes, one ancestor about 400 million years back. Predict the rest from this. A persistent pathogen gets classified by its reservoir and its clinical rhythm. Chickenpox, then decades later shingles, is a lytic burst, dormancy, reactivation, never a single clearing infection. Poliovirus is the book's eradication case study, and its load-bearing structure is a tool that turns on its user. 2 vaccines encode 1 trade-off. Salk's killed virus is safe but costly and needle-bound. Sabin's live attenuated virus is cheap, oral, and it sheds, buying gut immunity and passive herd coverage. The mass campaign needs the cheap and shedding option, so Sabin wins. But shed live virus reverts. In the endgame, the vaccine itself becomes the last reservoir, and the residual paralytic cases are vaccine-derived, so eradication must invert its own instrument, retire Sabine, return to Salk precisely as it nears zero. The generative pattern is this. Any self-propagating remedy becomes the disease's final refuge. This section explains the pox viruses and the single structural rule that defines them. The pox virus is a DNA virus that refuses the nucleus. Every other DNA virus borrows the host's nuclear machinery to transcribe itself. The pox virus brings its own. Its RNA polymerase is packed inside the Brit-shaped virion, so it replicates wholly in the cytoplasm, self-sufficient. Autonomy is the primitive. Carry your enzymes and escape the compartment that would constrain you. Genome size and structural complexity follow as consequences, not as additions. The chapter's anchor is variola, smallpox, the only human disease ever eradicated. It was undone by Jenner's cowpox trick, an immune system trained on a harmless cousin. That cousin, vaccinia, now ferries other antigens as a vector. Knowing the one rule broken, you can predict the whole family, a large genome, self-packaged transcription, and a cytoplasmic factory. This section explains why the poxvirus builds its life cycle where it does. Every other DNA virus commutes to the nucleus to borrow the cell's transcription machinery. The pox virus does not, because it brought its own. Packaged inside the virion sits a complete DNA-dependent RNA polymerase and about 20 enzymes, enough that replication runs entirely in the cytoplasm. Inside self-built factories, the virus walls off with barred membrane. The primitive is that autonomy dictates location. What a virus encodes rather than borrows sets how far it can drift from the host's machinery. The pox virus, near cellular in self-sufficiency, drifts furthest. It is the largest human virus, barely visible by light microscope. Chapter 15 pairs this with its counterweight, smallpox, the only human disease ever eradicated, and Jenner's cross-reactive cowpox as its lever, as the lever that did it. A virus never appears from nowhere. Every new one already circulates in some animal reservoir. What changes is not the virus, but the boundary between that reservoir and us. Lawton's chapter reframes apparent randomness as a contact surface event. Emergence is what happens when human activity newly touches or moves the interface with an existing host population. Read that way. The sprawling table of causes, urbanization, air travel, deforestation, dams, bushmeat, climate shift, collapses into one variable with many knobs. Each one widens or relocates the human to reservoir contact surface. Hence the two anchoring statistics. About 75% of these diseases are zoonoses, and RNA viruses dominate. Predict the next one by asking where a boundary is about to be crossed. A virus doesn't emerge by inventing itself. It emerges when the boundary between its reservoir and us opens. Bourbon virus was already in the tick. What changed was a man's shoulder. So emergence names, a coupling, not a genesis. 3 knobs turn it. The first is human behavior, widening contact, through travel, bushmeat, urban density. The second is ecology, dragging the reservoir into range, through deforestation and encroachment into wild habitat. The third is the genome itself mutating or reassorting onto wild habitat. 3/4 are zoonosis. The animal held the virus first. Emerging and re-emerging are one move. Seen twice, a closed interface opening. They differ only in whether the pathogen is new to us or merely returning.